From Microbe TV, this is Immune, episode 23, recorded on August 14th, 2019. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back.
How are you?
I am good. It has been really beautiful, and I had a couple weeks away, so I'm good.
And from Durham, North Carolina, Steph Langel.
Hi there. Good to be back. The amount of time between since we've last talked is shorter than the previous episode, so this is nice.
Yes, that's the vagaries of scheduling, you know.
I know.
The next gap could well be longer.
It might be.
Well, it might be. Did you go to Amsterdam? Was that where you were headed next, Vincent?
No,
the Netherlands.
I'm going next week. I'm going to Stockholm.
Oh, that's what it was.
Sunday for a week. I'm doing 3 TWIVs there in 3 different places.
Wow, 3 TWIVs. It's a lot.
Yeah, so I was originally invited to go to a meeting of the Swedish Society for Virology and do a very interesting TWIV with Erling Norby, all about virology Nobel Prizes, because he's been on committees that decide a lot of them. He's written a couple of books. And then I thought, well, I'm there, why don't I do some podcasts in Stockholm? So I had a couple of people I had met. I met one person in Rotterdam earlier this year, and she said, if you ever come to Stockholm, you know, we'd be happy to have you. So, hey, I'm coming to Stockholm. So she's arranged a podcast. She's at the Karolinska. There are multiple campuses, apparently. So she's at one, which is more clinically oriented. So it's kind of clinical virology. And then the other campus of the Karolinska, another TWIV listener, he said, I heard you're coming to Stockholm. So I hooked up with him. And so I'm doing 2 TWIVs. Yeah.
That now is one of those Peter Broden. Is that one of the TWIVs he's going to be on?
It's a good question. I don't know yet.
Well, I only ask because somebody had inboxed me from Karolinska telling me that he will be one of the panel members. At least that's what they said. And he's an excellent immunologist, systems immunology. We did one of his papers on children that are born premature and how their immune cells change over time. I'll have to look up which episode number, but yes, so I think that'll be a good immune crossover if he in fact is on the panel.
Yeah, Peter Broden.
Yes, and I was going to say maybe it's Petter. It's probably yes.
Petter
so I think
Broden and
that's going to be great.
Anna Smed Sorensen and Jan Albert. So
great.
Petter Broden. Apparently, we did his paper on TWIV 544. Oh, antiviral antibodies in the newborn. Yeah. So that's what he does. That's what he does.
Yep.
Okay, well, why don't you come with me and ask the question?
I was going to say, could I come?
You know, if I had money, I'd say, yeah, for sure.
I know. Do we want to crowdfund Cindy and I, immune listeners, to go?
You got to do it quickly because I'm leaving Sunday.
I know.
Although you could leave Monday because the podcast is on Tuesday.
That's true.
Oh, so do you know him? You know his work, I guess?
So, yeah, I do. And we have communicated back and forth. So the lab I'm in now and his lab are interested in very similar things, the immune development, both in the pregnant mother and the infant. So, yeah, he does great work, and I'm looking forward to that.
Well, I have to ask the right questions, right?
Well, yes, we could supply some.
Yeah, that'd be fine if you would.
Sure.
I'll pick a few of his papers and let you know, and then you can give me some things that I wouldn't normally ask, right?
Yeah, that sounds great.
And then, of course, he could say anything, and I wouldn't know if he's... No, I'm sure.
Oh, here we go. So I'm looking, it would have been Immune episode number 12, Systems Immunology.
Oh,
the title.
So we did a paper on also immune. Wow, two different podcasts.
Yes.
Oh, that's cool. That's cool.
Yeah. Mm-hmm.
So I have 2 podcasts on the next Tuesday. So in the morning is at one campus, and then the afternoon is at the other campus, and I have 3 guests on each one. So that'll be fun. And then Wednesday I go to this meeting, and then Friday evening we have the other podcast. So anyway, that was a long answer to whoever asked me where I went. So that's my August travel. And then,
Nice.
then in September, I'm going to Copenhagen.
Hmm.
Then I'm going to do 2 podcasts there. And then in October, a few in the U.S. And then in November is Germany for the giant virus meeting.
Hmm.
Wow.
And then,
So impressive sounding.
and
Giant.
then December, I just got invited at ASV to go to Singapore.
Oh, nice.
There is a Nipah virus meeting
Hmm.
there, and I'm going to speak with the people who discovered it and get the story. That'll be fun.
Yeah, that's great.
Have podcast will travel.
Exactly.
Cindy, do you have any other travel? I know you're in Germany.
Yeah, I was in Germany. Where am I going? No, in November, I go to the Society for Leukocyte Biology conference.
Where's
Oh,
that?
nice. Where's that?
That's going to be in Boston. and
Long
then
long trip for you, huh?
Yeah, no, not too far. But then I'm gearing up to go to AAI next year. So.
Oh, are you? Okay, so it is in Hawaii. I think we talked about
It
that
is
before.
in Hawaii. Yes.
If I end up there, Vincent, you should try to come out and then we do a 3-person podcast out there.
You know, when I was at ASV, a student from Hawaii came up to me and she said, "We want you to come to Hawaii to do a
Ooh.
podcast." And I said, "Yeah, I'd love to go because my wife would actually love to go on vacation, right? So I would take her with me. And I could possibly, when is this meeting?
It's in
May. It's always over
May?
Mother's Day weekend. Yes.
Which is, but hey, Vincent, I mean, your
Wow.
wife, Mother's Day gift, take her
Well,
to
listen, the
Hawaii.
kids are all grown up. They don't really want to travel with us anymore.
Yeah.
And frankly,
Not even to Hawaii.
I don't
You don't want
particularly
them.
want to travel with them either. They're in their 20s. They do different
Yeah,
things.
no.
And
Yeah.
first of all, the problem is they sleep too late for me.
Yes.
And the
Mine are starting to do that.
And the
too.
day is like half done before they, and I'm not, I'm up at 6. Forget it.
Yeah.
I think when they start getting in their 30s, they'll start coming back around for some, advice. I don't know. At least I found that. 20s, they go off, they do their own thing. 30s, you come back because you're,
My son had a party this weekend and all these 20-somethings in there. They're talking about sleeping late. And I said, I can't sleep late. And they said, no, don't tell us that. We're going to lose this ability. I said, yeah, at some point you will. That's life. Then
because you have a job mostly.
well, then you can do a lot more in the day, right?
Yes.
Although, you know what they do? They sleep late and then they stay up all night.
Yes.
It's crazy. It's
Can't
totally
handle that.
crazy. Are you going anywhere, Steph, this year?
Nope, not this year other than just holiday travel. But in terms of conferences, work we've done so far since I've arrived at Duke, I'll hit up some of I'm hoping for AAI, but there's a Keystone maternal neonatal immunology I might go. It's in Colorado. So we'll see. But 2020 for me will be more travel.
Well, I missed both of you at ASV because you were there last year, remember?
I know.
So I
Maybe
was-
we'll try this coming
I
year.
was in Minneapolis, and that was fun. Minneapolis is a nice place, and it was a great meeting as usual. Next year, it's in Fort Collins, Colorado.
Yeah. Mhm.
Ooh,
Possibility.
Yep.
I like Colorado.
It's a very nice state, yeah.
Yeah.
If you like what we do here on our podcast, please consider supporting us financially. We could use your help. Go to microbe.tv/contribute. We'll talk more about that later. And we have a follow-up email from Kim, who writes, Hi, Immune Team. I'm Kim, a fourth-year PhD student studying lung immunology at Boston University School of Medicine. I've been a fan of TWIV for years, but was so excited when Immune started. Thanks for venturing beyond your comfort zone, Vincent. I don't know if I am.
Well, listen, and beyond your comfort zone, speaking of papers that we're going to be doing today, I think each one of us goes a little bit outside our comfort zone for this.
It does.
Oh, yeah.
It does. I would love an excuse to go back and re-listen to transcribe all the prior episodes of Immune and provide transcripts for you moving forward as well. Please let me know if this would be welcome, as well as any helpful tips based on the TWIM transcripts being done. As always, thanks for your excellent work on the podcast. Best, Kim. P.S. I believe at least Vincent knows my older sister, Brianne. Brianne Barker.
Awesome.
Brianne's sister.
Kim Barker.
Yeah.
Well, that'd be great. I mean, we talked
Oh,
about
that would
that.
be-
Well, we actually talked about it on the last episode.
Yeah.
I think
Yeah.
because
some
someone
people have
wrote
been
in.
asking.
Someone wrote in.
Yeah.
So
Yeah.
Kim, I will be in touch with you. I will let you know how we do it for Immune for TWIM. There are actually some programs that you can now use to run the audio through and give you a first draft, and that can help you a lot. You'll still have to correct and annotate, but it might help.
Does it learn? Can you train it?
I think it does.
learn?
I think it does. Yeah.
because when I had broken my wrist, I used Dragon.
Yeah, Dragon learns.
And you have to teach it, but it
cool.
It did start to work really well.
I think they do. So I will be in touch with Kim. I wanted to read this before, and I'll email you, Kim, and we can talk about the details. But that would be great. Yeah, because there's not that many episodes, so you could definitely eventually get to them all.
Yeah, thanks, Kim.
Isn't that cool?
Yeah, thank you.
Okay, now it is Cindy's turn to inform us.
I will try. I'm going to need help on this one, so we'll keep it generally not in the weeds detail. But I picked a paper that I thought struck me as being incredibly important for all of immunology. But I will preface this by saying a lot of it is outside my comfort zone. It's a lot of genetics and genomics that I wasn't entirely familiar with, but I think the main points get across, and I think it'll be really exciting to talk about. So it was published in Immunity in July, so it's very, very recent. The title of the paper is Defining Genetic Variation in Widely Used Congenic and Back-Crossed Mouse Models Reveals Varied Regulation of Genes Important for Immune Responses. So the first author is Chisholm, and then there's Chang, Colborn, Silva Sanchez, Metza Perez, I don't know how you say that, Randall, and the senior author is Amy Weinman. And so the bottom line of this paper, if we lose you at any point, is that I say a mouse is not a mouse is not a mouse. Because what they found was that mice that we typically think are genetically identical are not. And so in immunology, we often use, and I'll talk about this in a little bit, we use mice that vary in one marker so that you can track cells of two different parental genotypes in a third mouse. And so we assume that they are genetically identical and they behave the same. And in fact, they don't. And I think the reason why this paper is really important is because I think it's conceptually going to make us think about where we purchase our mice from, how we annotate in our papers what genotype of mice we are using. And it may actually change some regulations as far as NIH or journals requiring you to put information in there about where you got your mice and what exactly they are. Because many of us know that if you make a transgenic or a knockout, you back cross, which means you breed to the parents or breed to a pure line for a long number of generations, and you keep screening to make sure that your altered gene is still there. And we assume that after, people say, 10 generations or so, it's pretty much pure. And what they find is that, in fact, is really, really not the case. So have you guys used genetically modified mice?
Yeah, sure. I remember we made transgenics. We made transgenics years ago, and
did you do this? Did you back cross?
No, we didn't back cross at all.
Transgenics is not quite as important, right?
I mean, we put the polio receptor into mice, and we were infecting. We didn't do any immunology whatsoever. But you could still argue that things could be different. But I just know that whenever we'd have litters, they would be all different colors because right? You'd have white ones and black ones and brown ones all the time. And
you're just like, oh, they're pretty.
Well, it's just because the one you use to make the transgenics are a hybrid to begin with.
Right. Yes.
And we didn't do any back crossing, and people didn't really have any issue. But if you're going to do some of the things like they do here, yeah, it's an issue. Yeah, I mean, even definitely. 10, I don't know why we assume that 10 would be enough,
I know.
Right?
That's what I thought. 10, I guess, is because your allele of interest is retained, and you still have the genes of whatever your recipient, your homozygous inbred mouse. But why 10? Did we decide?
I don't know.
I mean, it's a nice number, but I think as we get more advanced in what we can actually look at, and we'll talk a little bit about that in terms of like our next generation sequencing capabilities, we're learning a lot more. And I thought about what immunologists around the world were thinking when this paper dropped. It's just, I think it's going to really, like Cindy mentioned, change the way we have to just be way more careful, as if science wasn't more difficult enough. What our knockouts, what our transgenic animals are telling us, because if you're like, we're going to talk about buying them from different places, where they come from, what their background is, it's going to change your interpretation.
Well, we often get mice from people, right?
Yeah.
Go, let's try this
too.
knockout. The guy down the hall has it. So you get it and you do it. And now we shouldn't do that anymore because we don't know. But
you need to be careful.
As you're going to tell us, they have ways maybe that we can use to see what's going on.
Yeah. So, I think that when this, as Steph said, dropped, I think it was a collective
Everyone starts sweating.
Yes. But I mean, it's not necessarily new to immunology,
right?
Because I think one of the first big uh-ohs that we found was there was a paper that showed that there was a difference in microbiome, especially related to one particular bacterium called segmented filamentous bacteria, or SFB. And when that paper came out and we discovered that SFB was different, it was in the same black 6 mice, for example, from one vendor had it and another vendor didn't, and it influenced the immune response in the gut. So we kind of already have this to some extent. We also have ideas that microbiomes can be different. So mice housed conventionally or in a germ-free facility or in specific pathogen-free rooms at different institutions have different microbial components. And so we often, for example, acclimate the mice for at least a week. And if we're going to do immunology studies, infection studies, we'll often share bedding or things to make sure that they have equivalent microbiomes. So that's not a factor altering the data. So if you've got 2 cages in 2 different rooms of your knockouts in one room and your wild types in another room, for example, if they're have different microbiomes, you could actually have different results for immune outcome that aren't necessarily dependent on the genotype difference.
We have a guy here who his whole thing was when he was a postdoc, he found, and this was in Dan Littman's lab. Yep. He had these 2 mice, same strain, one from different breeders. One had this bacteria in the gut and the other didn't.
Right.
Segmented filamentous bacteria, which made a big difference in the development.
Yep.
Right.
But
Iliev, right?
Yeah. Yeah. What's his name? I don't even know the names of my colleagues. Ivo Ivaliov or something like that.
Yeah.
He's Romanian. But the thing is, we can do all this, but mice are nocturnal and we're making them live during the day,
So we covered that on Immune too, right?
Yes.
I know, it's just that the bottom, beneath all this, we're totally screwing them up and nobody ever talks about it, right? It's just for our convenience so we can go in during the day and do our thing, right?
And the temperature. Right. We keep the temperature at a good temperature for humans, but not for mice. So so we have to be cognizant of this that we're going to go over in this paper. But in reality, we do the best we can with what we have. Mm-hmm. And I think that the thing we always have to keep in mind as researchers is just say what you did. You do what you did. You made a choice for a particular reason. As long as you outline in detail what you did, then we can interpret based on that.
They say something
because there's never a perfect
in the discussion, right? They say all experiments are imperfect.
Right. It's true. It's right.
That's
right.
It. That's basically it. So you're right. You have the good. You have the moderated view. There's some people, of course, who say all mouse work is crap.
Well, that's
right.
Yeah.
That's the opposite.
Yeah, I mean.
And there are people, and you can read articles by them in journals who say all this is useless because of this and this and this, and that's the other extreme.
Well, yes, if you're planning on taking something into a human immediately, it's not going to be highly relevant. But we learn a lot from this.
No, totally. You have to do things in mice and other animal models for sure. And then, as you say, you can't say this is it. You have to further test them. Mice lie and monkeys exaggerate. That's what we say.
Yes.
Yes.
That's what we say.
Yeah, exactly.
And a lot of the historical fundamental immunology discoveries, histocompatibility, MHC restriction, thymic education, it was all
Yeah, sure.
using mouse models like this. But of course, coming from a background where I use pigs, I see the other side where if you have only homozygous inbred mouse strains, what is that really telling you? And so I like this paper because it kind of talks to the limitations of mice. But of course, this paper offers us a way to, what Cindy's saying, recognize your limitations of your model so you can report that.
So getting into the background for this, do you guys have hobbies?
Vincent, do you have hobbies other than podcasting?
I don't. I mean,
I like
there's nothing I do at home. When I go home, I read about viruses. But no, I don't have hobbies. I consider, science is my hobby.
Well, I have a couple of dogs. I really love state parks, so I enjoy hiking. I enjoy anything outdoors. So that's one thing. There's a couple others, but what about you, Cindy?
Yeah, I try to exercise, but I read about vaccines, I guess.
Yeah.
I mean, I cut the grass, but it's not a hobby. It's something I have to do.
No, that's sort of a torture, right?
Well, I don't mind.
Some people think it's meditative.
It's the only exercise I get, but I do get to think a lot while I'm.
Yes,
for sure, exactly. I get to think uninterrupted. But
anyway, there was a purpose to this, and that is that, did you know that we wouldn't have any of these mice if there wasn't a woman who, or a number of people whose hobby it was to breed mice?
Yes, that's correct.
Someone brought them over from Europe at one point.
They did. So in the 17th century, Japan, it was actually a aristocratic hobby to breed fancy mice.
Fancy
mice. And so there was this, the history of this is there was this woman, Abby Lathrop, who lived in Massachusetts, and she had a farm and a barn where she bred lots of different things, and ferrets and rabbits and mice. And she had over 10,000 mice that she raised in her barn.
Oh my God.
Can you imagine?
Were they in cages or just running around?
I think they were in cages to some extent, right? Yeah. But anyway, so the story goes that she was a very savvy businesswoman, and she would sell these mice to people who liked fancy mice. And there happened to be an individual at Harvard who wanted to study short lifespan. So he was actually a researcher. His name was William Ernest Castle. And so he got these mice from her, and his grad student was tasked with inbreeding some mice to see if they could generate a pure line. And so this grad student's name was C.C. Little, and C.C. Little created the DBA mouse. So have you heard of the DBA mouse? So it's a dilute brown non-agouti, is just the color coat. And so it's the oldest inbred line known. It's now known as DBA2.
That's cool.
Yeah. So, but when this guy started inbreeding these mice, Lathrop said, maybe I should inbreed mice too. So she liked black coat colors. So she bred and came up with the C57 black mouse. So that's the standard mouse that most people use in their research.
There's a story where Swiss mice came from Switzerland. Some lady brought them over in her dress or something, right?
Or something like that. Maybe I missed that one, but that's a cool story.
Interesting.
Too. Yeah.
Imagine it came over on a boat, I guess, but she had them in her dress for the whole trip.
Oh, no, thank you.
Oh my gosh.
I have lots of questions about that.
I don't think I want to do that. I'm not like squirmish of mice, but anyway, I don't know about keeping mice in my dress or 10,000 in my barn or a garage or whatever. But anyway, so she inbred these mice, and she came up with this C57 breeder stock, and she worked with someone at Penn named Loeb or something. And anyway, he decided to start a business, and that is now the Jackson Lab.
Very cool.
And so they have over 7,000 different strains of mice. So I'm sure they have many more than 10,000 mice with 7,000 strains. But yeah, so a lot of these mouse strains are derived from those original ones. So here's a question for you. Can you guess how many mice are used in research every year?
Whew.
You mean globally or just?
Yep, globally.
Oh my gosh.
No,
every year? Millions, I would imagine.
90 million.
Yeah.
90 million.
We have to be very thankful for the mouse.
Yeah.
It does a lot for us.
And it really does a lot for us. And Steph, you mentioned a few things, but really it's been fundamental in helping us dissect and uncover and understand the immune system. You can knock out different cells, you can knock out different genes and see how you can challenge them with bacteria and viruses, and we can determine the role that all these different genes play in host defense. We understand antigen presentation, we understand vaccines. All of these have been amazing things that we have discovered by using mice.
And you can add genes, you can add transgenes.
You can add genes,
right.
you can
make
take
virus
away.
models for viral and bacterial and fungal infections. Yeah.
Right. And so one of the things that immunologists do is they make bone marrow chimeras. And I believe we've talked about this before, but generally you take and wipe out the immune system in one animal and you replace it with the stem cells from another animal. That will repopulate. You put hematopoietic stem cells, and that will repopulate with, deficient in a certain gene or a marker in a certain gene or whatever. And another thing that we tend to do is not only just make a bone marrow chimera, but make a mixed bone marrow chimera in which you can track, because maybe the knockout cell and the wild type cell behave differently when you put them in together versus on their own. And so in order to be able to pull back out those individual genotypes when you've made this mixed bone marrow chimera, we use markers that can differentiate between the two. And so these are called congenic mice or congenic markers. And a common one is CD45. And so that's basically marks all lymphocytes or all immune cells. And so they can be CD45.1 or 0.2. And so we have antibodies that can distinguish those isoforms of CD45. So you can mix wild type cells that are CD45.1 with knockout cells that are CD45.2, mix them together and put them into a mouse in which you've wiped out the immune system. And then you can look at how those different cells perform in that context. However, we're going to find out that maybe that's not as easy as one would think, which is related to this genetics.
The basis behind that was that although, they were detected by different antibodies, they really behave the same. That was the assumption behind these two different types of mice strains, congenic mice.
Right. And the assumption is that that's the only place they're different.
Right.
Except for what you've messed up. So if you have a wild type and a knockout, the only thing that's different is this marker so that you can functionally compare them. So, yeah. So the other thing you need to know is about how knockouts and transgenics are made. So knockouts, you usually make them in one background because they're particularly amenable to knockout for whatever reason. We have a 129, which is one of these strains of mice like DBA or Black Cyst or Balbc. So these ES cells come from 129. You knock out the gene, both alleles in those cells, and then you put them into a blastocyst and it grows up into a mouse and you breed and you back cross. And typically we back cross to a Black 6 because most of the knockout strains, for whatever reason, I don't know why we didn't keep it on 129 to begin with, but we make them and we back breed them to Black 6. So whenever you get a knockout, you need to check, but probably 80 to 90% of the time they're on the Black 6 background. And so you can then compare knockouts and wild types on Black 6 and use the Black 6 as a control wild type, et cetera, et cetera. So that's how those are made. The other thing that you need to know is with microbiome studies, and we have, we mentioned SFB, but that's not the only thing. Different microbiomes can cause inflammation or be protective or whatever. And the way people study this is they derive mice into a germ-free environment. So basically, they take pregnant mice and they give them a C-section into a completely clean environment so those mice grow up. excuse me, without any bacteria at all. So none on the skin, none on the fur, none in the gut, none in the lungs, nowhere. And they use these to try and understand what the bacteria are doing to develop different things and how these mice might have deficiencies, et cetera, et cetera. And often what people will do is they will co-house these germ-free mice with wild type mice with their own microbiome. And then those germ-free mice will pick up that microbiome. And it's thought that the microbiome secretes various different things that can modulate immune responses. So that's an important concept that'll come up at the very beginning, because when they tried to do this, it revealed that there were differences that were probably not microbiome mediated. Right. That led them down this path for this particular paper. So the last thing I really want to introduce is ChIP-seq. So there's this process called chromatin immunoprecipitation, where you can immunoprecipitate chromatin based on markers that are on histones, and then you can sequence what's associated with that. And so what people have done is they've used this kind of approach to ask where transcription factors are sitting on the genome, or where the genome is marked with nucleosomes that have modifications that indicate that they're active. So it's actively transcribed. And so you can look at how the epigenetics, which is those modifications of the nucleosomes, are different between different strains of mice. And it's extremely sensitive system. And you can scan across the entire genome because you can sequence all these different fragments, and you can find out where these differences are. And so, I sort of come back to saying there's a lot of assumptions that we make based on reagents that we use, including mice. And so we have known knowns and known unknowns, and unknown knowns and unknown unknowns. And so trying to limit the number of boxes that are black to you, obviously unknown unknowns are always unknown. But now we know that our genetics might be different in our mice that we think are genetically identical. So it just raises the idea that we need to think more carefully about how we use mice, and what, if you do see a difference, what it's actually due to.
Right. And there was one of our previous episodes kind of touched on this, although we didn't directly bring it up, but I will go back. But I think it was talking when we were about pure apoptosis, and we were talking about caspases. And in that paper, they were using caspase 11 knockout mice, and
That's
really...
right.
Right. And because of this issue where this original embryonic stem cells from this 129 strain, it knocked out caspase 1. And so what that did in these early studies of caspase was it really mischaracterized the role of that protein in innate immunity. So if you want to go back and listen to that episode and with this one, it's kind of a nice way to look at it in action. But I think, again, Cindy, these assumptions are huge. And the reason I think they're being challenged is because now we have the technology to look at it like this paper does.
Right. And so if I look back at these kinds of studies, I wouldn't say necessarily that they were wrong. They were right with the information and the technology and the tools that they had at the time.
Yes.
But now we have different ways of evaluating this, and we reveal things that we didn't know before. And I think that's important, because you don't want to blame people for making incorrect evaluations or invalidating a whole set of literature. It's just that now we know more, and so we need to go back and reevaluate what we thought. And I think that applies to all areas of life too.
That's true. And what it will do is just make things, you just would have to be more rigorous and more thorough to confirm that your model is actually revealing what you are looking to reveal and what your hypothesis is.
That's right. So what this paper originally set out to do is they wanted to ask if the microbiome affected T cell function. So why might one think this? Well, I mentioned it. The microbiome is all the bacteria that live in your gut and elsewhere on your body, or the body of a mouse. And they can secrete, we now know they can secrete molecules that can go into the bloodstream, and they can affect cells and the function of cells. And so one of these metabolites is short chain fatty acids, and they've been shown to play a role in a number of different things. And so they really wanted to know, did this play a role in T cells and T cell function? And so to start, they isolated CD4 and CD8 T cells from conventionally housed or germ-free mice, which is exactly what anybody would do to do this study. So with bugs, without bugs. They take them in vitro, polarize them to the Th1 or TC1, and then they looked at these markers of activity on the nucleosomes. It's called histone 3, lysine 27 acetylation. So you have an antibody to that, you pull it down, and then you sequence, and you find out where in the genome do they have these marks. And you can look all across the genome. And what they found were differences. So they were like, awesome, microbiome is affecting T cells. So they wanted to confirm this, so they confirmed it by PCR. These differences were all good. That's great. So what do you do next is exactly what I said. You co-house the germ-free mice with the conventional mice, and they are going to transfer the microbiome, and then voila, the T cells should behave just like the conventional mice and not the germ-free mice. And the marker should be the same. And guess what? They weren't.
Wah, wah, wah.
Right. So one could stop there and say, okay, well, it's not due to the microbiome. So what is it? And so their first clue was, I guess they tasked someone with mapping where the differences were. And it turns out that almost every difference they saw was on chromosome 1. And so that would be unexpected. I don't know a lot about genomics and genetics, but I would think that those differences should be spewed throughout across the genome. And they all seem to be on chromosome 1. And they said, that's kind of weird. And so they went and looked at the backgrounds. I don't know how they would have figured this out, because it wasn't something that I would have thought of. But eventually they figured out that their germ-free mice that they bought and were housing had the CD45.1 marker, and the conventional mice had the CD45.2 marker. So they said, maybe the genes surrounding this on chromosome 1 are different because there's a small chunk of the genome that's left in the one strain of mice when they backcross to make this congenic strain. So they retained some DNA from the CD45.1 background, which was called SLJ, in the germ-free mice, and that's why they're different. And so who cares, right? So it's a small chunk of the genome that's retained from a different strain of mice. Why would that make any difference? Well, turns out that particular part of the genome encodes a whole bunch of genes related to immune function.
Yeah.
So that's a big oops. So when they were looking at congenic markers to look at differences between immune cells, they chose this it's a lymphocyte marker. If anybody looks for any immune cell, if they look for an immune cell, they use CD45. That's the marker that tells you it's an immune cell, right? And it turns out that, of course, that's in an area where there's a whole bunch of immune genes that are encoded. So they said, okay, well, one of these genes is cathepsin E. And guess what cathepsin E does? It's important in chopping up antigens for presentation on MHC. So this could actually have some
pretty
real
fundamental
implications in what we're doing. So why is this expression different? So what is it about this region that might be different? So first of all, they looked at cathepsin E. They showed it was actually differentially expressed in the 45.1s and the 45.2s. And when they explored it by looking at the sequencing, it turns out there was a long terminal repeat inserted into one of the congenic ones, a CD45 or 1 or 2, and that actually changed the expression level of the gene of cathepsin E.
Vincent, were you excited when you saw LTRs?
Well,
did your ears poke up?
So I don't know why they think it's viral, because there are lots of LTRs in genome that jump around. They're not. They're transposon based, right?
They were originally,
No, they weren't. They were LTRs before retroviruses. Many people get this wrong all the time. They say it's viral. So there were retrotransposons, retrotransposons containing reverse transcriptase with LTR before retroviruses. They gave rise to retroviruses when they acquired an envelope glycoprotein to allow them to make particles. But long before these viruses were circulating, our genomes had these transposable elements that had everything that a retrovirus has except for the glycoprotein and the capsid protein. So
I guess that's a misnomer.
So they say integration of a viral long terminal repeat.
That's right.
And they say 7.5 kb in length. Now, first of all, no LTR is 7.5 kb in length. So I don't know what they mean by the LTR is less than a kb. It's only a piece of the transposon.
Yeah.
I suspect this is a transposon because they don't tell us why they think it's viral. What would make it viral would be to have a glycoprotein gene, which is clear. Mice have all the time and they jump around, but transposons can jump around. So I suspect that's what happened. It just Yeah, so I thought that was cool, but it's not surprising because these, whether it's a endogenous retrovirus or a transposon, they always move around the genome, and you can imagine it's going to be different from different mouse strains, right?
Yeah, fair enough.
So the bottom line is this stuck into the gene and it inactivated it, right?
That's right. Or lower the expression level, knocked it out in some cells. And so one might say, well, okay, but does that really affect anything, widespread in all of the different types of mice that we use? So they went and they screened mice and they looked for expression of this cathepsin E. So was it at high level or low or lost? And they looked at whether or not by PCR they had this LTR, which we'll just call an LTR. We won't call a viral LTR or anything else, transposable element. And what happened was anytime that the strain of mouse had that insertion, they had very little expression of cathepsin E. So it was a 1-to-1 correlation. So it just suggests that in some strains of mice, they have this transposable element that popped into the gene and affects expression. So one might want to know, well, how widespread is this? And could we possibly ever look at this? And so what they did was, and this is supposed to be the crux of the paper, which I think most of the immunologists will gloss over, but they created a method, a computer-based method to screen to see how frequently this type of thing happens. And for those of you out there who are into genetics and genomics, this is going to be way too simple because I'm not sure I actually understand it. But basically, what they used was a little secret. It's the genomics dirty little secret is, I guess, the way. And I actually loved that because it was the immunologist's dirty little secret that gave us an immunity, But anyway, the whole point was that the way they align reads in these next-gen sequencing platforms is they give some flexibility because there are errors in sequencing and so forth. So they allow it to have a little wiggle room. And so what happens is you can get annealing where it's not exactly right. And so you think that all of these strains of mice might have the same sequences when they might not. And so what they asked was, could they use a more stringent approach so that they could pick up when these sequences were there in one strain and not in another? And so they did it two different ways. They inspected the sequences and did a computer-based approach. And then they also did a visual approach, because I guess it can visualize the sequences and you can scan across and you look for areas in which they don't align perfectly. So that's, if you guys understand it any differently than that, please speak up because that's about my extent of my knowledge of how they did this.
That's exactly right. That's perfect. Yeah.
That's the way I read it. And it's really, it's very, simple but ingenious because you can then see the differences between that reference, what they're referencing within the software and make it more strict and then see what's left out. That's right. It's simple, but it's great.
So the argument they make is that you can retroactively go and look at all of the RNA-seq data that are out there and use a similar approach to find out what genes are different in different strains of mice in a particular cell type.
You should probably tell us what RNA-seq data are.
Yeah, so it's RNA sequencing. So you take all of the RNA from a cell, you put barcodes on it, and then you sequence it en masse, and that tells you what that particular cell population was making at any point in time.
Yeah, it's the total RNA of the cell, right?
That's right. So when they did this, they identified the region that was different between these different strains, and they looked at specific immune genes that might be different, and they focused on one that's called CXCR4, and you might know that because it's an HIV receptor. Mm-hmm. It's also important for a bunch of different things, including recruiting cells to the lymph node and so forth. So this is where it gets really interesting, because now they said, okay, well, let's buy black 6 mice or whatever the background was. It's B6.SLJ. CD45.1 in case you're
SJL.
An aficionado. What did I say?
SLJ.
Oh, SJL. Sorry, I typed it backwards. But so they bought those same mice. So it's the same. We're not talking CD45.1 and 45.2. We're talking about the same exact strain of mice from Jackson, Taconic, or Charles River. And they asked, do they have the same expression of the CXCR4 and are there differences? And that's really kind of scary because they were different.
They have a beautiful graphic way of displaying it, right?
They do.
It's lovely. You can just look at colors. You can look at colors and you can see.
Yeah. And you can see that they're not the same, which is kind of scary, I think, because it's one thing if you think you've bred some mice and you assume that they're the same, but if you get a black 6 mouse from one vendor or another vendor, you would hope that they're the same.
Right.
So when you say you hope they're the same, what do you actually mean? You'd say that
they're genetically identical.
Well, of course, that's never the case anyway, right? Because there's always
They can get mutations. That's right.
I mean, even within us or within any multicellular organism, every cell has a different DNA sequence, right? Because that's just because that's DNA is error prone. But so when you say identical, and how identical are you talking about? Of course, here they're not close, because if you just look at these patterns, you can see there are lots of differences. So there
are lots of differences. One would hope there aren't that many differences, right? Yeah. So yes, you can have genetic differences, but how much of your genome is actually exons? And when you have a mutation, how many actually end up in a non-synonymous mutation and of those, how many would change function? Right. So it's not that frequent, I would think, that if you have the identical strain of mice that you are breeding at 2 different facilities over time, that they should be that different. You can have mutations that change things. So for example, a strain that I am interested in, which is the C3H HE mice, because HEJ and HEN, one of them has a mutation in TLR4. And that's actually one of the ways in which they mapped and identified the LPS receptor, was using that difference. And so like HEN is from NIH originally and HEJ is from Jackson. So sometimes you can find what the origin of a mouse strain is. But generally, if you've got a SLJ CD45.1 on a black 6 background, you would tend to assume that there are few, if any, genome differences that result in a difference in amino acid sequence that also results in a change in function.
I think in this picture, in this image on figure 4, which is a heat map of the differences, I think at that level, which is a 10,000 foot level, or maybe 2,000 feet, I don't know. It's not a nucleotide by nucleotide level.
Right.
There, you would probably expect that Jackson, Taconic, and Charles River would have really the same coloring patterns.
Right.
And, because the individual changes you wouldn't see unless there were a lot of them. So I think that's probably the metric when you would say we would hope the mice are identical, right?
Yeah.
Because they're not going to have the same DNA sequence. It's never going to happen.
Right.
Because just in the breeding there will be changes. But at this high level that they get in this visualization, I think it should,
at the transcriptional and protein level.
Yeah.
Right.
Yeah. But I guess, in the end, there were a lot more differences than one would have anticipated. And what they found was that in some of the mice, so on chromosome 1, they saw differences around this the CD45 locus in some of the mice, but then on another mouse, differences were on a different chromosome. So it's not just related to where the CD45 gene is inserted. So there's more widespread differences among different mice. They should be the same mouse strain across different vendors.
Right.
So are these differences on multiple chromosomes or on one chromosome? Because you mentioned one before.
So one is on chromosome 1, and some of the mice, some of the strains have differences on chromosome 19, and one of them has difference on chromosome 7.
And other chromosomes are similar, right?
Yeah.
So I would have liked to see the similar ones. Just even in supplementary
for
data,
reference.
Just say this is what we would expect the mice to look like, right?
So I guess in the heat map that they show, all the chromosomes are there. Not all of them, but some of them are there.
Yeah, they have.
I guess they only show the ones that have the bigger differences, but
Yeah, well, I can't really tell because you're right. On the right, they have chromosome 1, 10, 11, 13, right? Well,
Yeah.
but I don't understand the color. Where where that's coming from, unless that bar on the left. I'm sorry for our listeners that we're doing this. But
I know.
Did you see the bar on the left in panel A? That range of colors.
That tells you which chromosome.
Maybe that tells you what chromosome. So they looked at quite a few, but not all of them, right?
Not all, right?
Yeah, not all of them. In the end, this is really important because these are not just any gene. These are often the differences are in immune-related genes. So we talked about cathepsin E is one that's important in antigen processing and presentation. Talked about CXCR4, which will be important for some viral infections. It's important for recruitment of cells to lymph nodes and so forth. And then there's also CD62 ligand, which is a key one in neutrophil rolling and transmigration. So those genes alone, if they were different, is going to completely change the immune capacity of the mouse. So that's a big difference. And when they looked at 40 different genes that were differentially expressed among these that were immune related, there were at least 20 that were twofold different in what should have been identical mice from different vendors. So when these are immune related, it really could potentially, affect more widespread the functionality. So how do you look at function? Well, in my opinion, this was the downfall of the thing. I got all excited because I was like, oh, they're looking at flu. One, Vincent will love this. And two, they're going to show us that it's functionally different. And the bottom line of these experiments are that they did look at T follicular helper cells, which express CXCR4, and it's important for their functionality and for antibody production and for, just for B cells in general in germinal centers and lymph nodes. And so they already know that it's differentially expressed. And so they look in flu and they challenge them, but they don't actually see whether they're protected. They don't look at the viral lows. They don't really look at much of any of that, but they do show that the flu induces T follicular helper expression of CXCR4, and that the ones that have the genetic difference fail to upregulate that. And they suggest that that's evidence that there's differential function in the immune system. And
this section drove me crazy because if you have already challenged them, I'm assuming they weren't different.
Probably
and
not,
that's and that's
Probably
why. Not. And so that kind of brings me back to a couple of the things they brought up. For instance, when you talk about cathepsin E, it's important in antigen presentation. Well, maybe why people really haven't either noticed this or had any concern about it is because it doesn't have a function difference that you can measure between the two. So maybe look at antigen presentation and see if the function of that transcript is affected. Because I think it's one thing to look at, I think they did transcript and protein levels, but if they're not doing function, I think that was the lacking part of this paper.
So I guess means that depending on what you're looking at, these differences may or may not make a difference, right?
That's right.
Yeah, right. And maybe there's some compensatory mechanisms that allow those differences to be, it's fine. And that's a lot to ask, right? This was a very hardy paper. If we were to ask them to do functional studies on all of these different things they found that were different, it's for another paper, I'm sure. But I think, what you mentioned, Cindy, the flu, it was a little disappointing, but it's surely still important. And I don't know if they could have maybe tried a different virus. They tried flu, but did they say why?
No.
Maybe HIV if CXCR4 is implicated.
They say because it's known that CXCR4 can play a role in homing in this infection, but
right.
I guess it's been shown for that. That's not.
But then we would need to see like homing data, or
Yeah, I feel like they chose an assay which they know should be different to
sure,
point.
Right?
But right. My thought on this was these people were really genomics people, and they discovered this method and they found it was related to immune genes, and they tried to throw something immune related to it, so that they could validate what they were doing. So they probably don't have the expertise for that. But they could have collaborated with someone who did have the expertise.
Sure. But, maybe
they are.
They say maybe they are, and they just, you can keep pumping out data and, collaborate, but eventually you got to just get the paper out. So that could have been it. Could have been it too.
Yeah. Yeah. So, that's one example. And what they decided to do was do another example. So if I understood this experiment correctly, what they did was they had TBX21, which is the Tibet gene, which is important for Th1 differentiation, which this is what they started with was, does the microbiome affect Th1 cells through metabolites was their original question. And so they're still interested in Th1 cells. And so the knockouts for the TBX21 gene were made on 129 and back-crossed a bunch of different generations. And then they compared them that were back-crossed to the black 6 on the CD45.2 background to CD45.2 black 6 that were not TBX2 deficient. So in this case, you're looking at a specific knockout. This is different from what they were doing before. They were just saying before, if you've got a black 6 or you've got a BALB-C or you've got a, black 6 from Taconic or Jackson, how are they different? This is these were a knockout that was specifically made to assess immune function and has been used on 45.1 and 45.2 background in mixed bone marrow chimeras to evaluate function of Tibet intrinsic activity. Right. Mm-hmm. And so they compared these to the CD45.2, and what they found were there were a lot of differences, and they were related to immune genes that are kind of really important. Things like CCR7, which is a chemokine that's important for recruiting cells, and then cyclin-dependent kinases and a bunch of other things, that they were actually non-synonymous mutations that affected protein function, and they confirmed the differences. And so what they were saying here was that even if you back-cross, which you think should create what's considered a congenic strain, so you've got your control mice and you've got your knockout mice, then there are still differences, and they can depend on the vendor. They can depend on how much you've back-crossed. And the last thing they said was that even littermates might not be the best control. And I was like, what can you possibly do? Because the littermates might actually have different genetics around the knockout than the knockout.
Yeah, sure.
I don't even know what you do at that point. What do you do?
This is what I was going to ask. So they've developed a method where you could look at sequence, RNA-seq data, and say, the mice I'm using, are there differences that could affect my results, right? Right. So that they say it's important for immunologists to have the ability to assess whether genetic variation might be a relevant variable, right? So that's one thing. Or you could just rederive all the mice and make them identical or as identical as you can, so you don't have differences in littermates. That's crazy, right?
Right. So I guess the argument would be to use CRISPR, right, to knock out specifically your one gene, but keep it on the same background.
Exactly. But the thing is, whatever you're doing with your particular mouse strain from Jack's may be different from somebody else's, which is from Taconic, and they've done the same exact CRISPR thing, and they're getting different results.
Right.
So it seems to me the best thing to do would be for everyone, if you want to use your mice, C3H, you said? Everybody has to have the same C3H. Start with the same C3H. And Jackson and Charles River and Taconic are going to have to harmonize. That would seem to make more sense than looking at RNA-seq data for every mouse.
Yeah.
That's crazy.
I mean, that would be the most logical, but can you imagine trying to tell those three, giant companies that they have to have the same? I mean, I guess they could, but as long as they could still make money from it, I think that would be a challenge.
Yes, I agree.
You can't make that.
They'd have to say, okay, we're going to keep your black 6, we're going to keep your, BALB-C, and they'd have to go down the line and say which ones they were going to keep from the 3, and then the other 2 would have to call their entire colony.
I mean,
right?
It would be insane.
And then rederive. So let's say, for example, we decide to have Jackson's Black 6 stay. So all the other vendors have to sack all their Black 6 mice and get the breeders from Jackson and start their own colony again. That's going to massively economically
that would be
eviscerate those companies for a long time.
But how do you know which one is the right one, though?
Exactly.
Right.
Well, there is no right one. They're all artificial.
They're all wrong, right?
I think Jackson would say that they were the originals, so that they're the correct ones.
I think they have some legs to stand on there based on our original story, right?
Yep. Mm-hmm. Based on
but I don't know. I'm at a loss on how one conceptually interprets all these data and what you're actually going to do with them as an immunologist in the lab.
They don't really give you any help. All they say is you're going to have to sequence your mice, essentially, right?
Yeah, I mean, they give you this tool, exactly. Bioinformatics tool, but
I think that's not terribly practical, and it still makes it hard for labs to compare results because you don't know how, even if you have differences, you don't know how they interact and how they would change your results from lab to lab.
Right.
It's best to have the same mice, but I think that's really hard also. So I don't know what the solution is.
Well, really, I think
go ahead.
Well, I was going to say, I think all of this would take labs a lot more time to do research. And so there's not going to be an innate driver to do this. And so it would have to come down, I think, from the funding agencies that you have to do X, Y, Z. Because otherwise I don't think labs will take, I mean, thorough labs would, of course, but I don't know. I think it would just have to come from the top down if it's going to be something, consistent.
I think it would divide scientists because there are some that could be able to afford to do that.
Exactly.
It would be challenging.
Yes, totally.
So it will divide.
What I wanted to mention. It's already expensive to do mouse work. This is going to make it more expensive because you're going to have RNA-seq data now. Here's the mouse strain we used, and here are the RNA-seq data showing the genes in the area of what we were looking at, whatever. I guess you have to do the whole genome. You can't just do one chromosome, right?
Right.
So I wanted to bring up something else that's kind of related but very different, and that is, and I think we should do a paper that illustrates this in the future, and that is another kind of mouse called the collaborative cross, which I think both of you heard about, right? So here in infectious diseases, when we infect all these lab mice that Cindy's been mentioning today, these are highly inbred, even though they're different, they're still highly inbred. And they don't even come close to representing the genetic diversity of humans. And so you could buy all the different mouse strains you want, and sometimes your virus won't infect them because it's not the right genome. And so the collaborative cross, what they have done is taken 1,000 inbred mouse lines and crossed them all in a very specific way. It's an 8-way funnel breeding design, which they call it. And then you get essentially incredible genetic diversity that begins to approach that of humans. And then you can take these mice and you can start with a certain number. And let's say I have a virus that doesn't infect Black 6 or 129 or C3H mice, right? I can find a collaborative cross mouse that will be infected by that virus. Because it's got some gene in it that isn't changed in a certain way in all the other strains. And then you can map it. You can map the susceptibility and eventually figure out genes that allow viruses to replicate in mice. So this was done with Ebolavirus, which you have to adapt to these inbred mice to get it to replicate at all. And they found a series of collaborative cross mice that range from being infected with Ebola and nothing happens, all the way to hemorrhagic fever that completely mimics what happens in people. And you can map the genes. They're called quantitative trait loci.
Trait loci,
QTLs.
Yep.
And eventually, if you're lucky, you can get it down to a single gene and say, why does this allow the virus to replicate? Is it a receptor? Is it an immune gene or something else? So that's the advantage of this is outbreeding, really.
It is.
They took all the available mouse lines and they just mix them all together and you get outbred mice. And for certain experiments, that's good. For viruses, that's good because it allows us. So, we'd like to use that to make a mouse model for a virus for which there is none.
Right.
And
Right.
so it takes advantage of diversity. Whereas for immunology, you want it to be harmonized, right? You want the mice to be all the same, although we learned today that they're not really. So collaborative cross takes the opposite approach, and which I think is an interesting contrast. Right?
Mm-hmm.
Well, no, it's great. And we're going to have to start thinking more, heterogeneously, because you can really just type mouse research and find that there's a lot of struggles to translate it into human applications. And of course, we always talk about, we learn a lot mechanistically from our, mouse studies or in vitro studies. But if we have to consider the variability as the collaborative cross does. That's great.
I guess the extreme of that is people using the pet shop mice or the outbred mice, right? Wild mice.
Yeah. Or another animal model like pigs.
Yep. Yeah.
Anything else?
Wow.
Anything else?
I think
I know it's a bear of a paper, but I think
that's a good. No, I think it was.
I broke it down.
It's a good job. Very good.
Oh,
and then the thing is, we don't know what's going to happen. We're waiting to see.
Right.
The impact this has. It will take a while, probably.
Should we do email?
Yeah, let's do some email. That first one is written to you, Cindy.
Okay. Dear Cindy, this is anonymous, by the way. I'm writing at the recommendation of my friend who studies HIV vaccines as a postdoc. Some background. I'm a trained engineer, an MS in mechanical engineering, and someone who likes to dive into details. As such, I have found it immensely frustrating as a new, admittedly anxious new mother wading through information related to vaccines, because when I spot apparent inconsistencies or have other unanswered questions, I'm unable to find the answers I need to assuage my concerns without doing a full-blown literature review. The questions below represent just 2 examples of those types of concerns. So I can completely understand how you feel. It's overwhelming. There's a lot of misinformation. It's hard to find things from reputable sites, especially about vaccines. And I hear you about being a new mother. You just want what's best for your baby. You can't do anything else. Anyway, so the first point here is people talk about the amount of aluminum received over the course of the first 6 months of a baby's life in food and breast milk versus vaccines. However, the vaccines represent a higher intensity dosage of aluminum rather than a more prolonged exposure over time. Does the intensity of aluminum administered have an impact on its toxicity, especially as it relates to the blood-brain barrier? Are there credible studies addressing that topic? Do we want to do that first, and then we can address the second issue?
Yeah, sure.
So, Vincent, you had some interesting information. I think one of the first things we need to clarify is it's not aluminum. So we're not talking about like aluminum foil that you wrap food in.
Right.
It's alum, so it's salts of aluminum that are precipitated.
Right.
And you found a source that says that they are either aluminum sulfate mixed with sodium or potassium hydroxide.
Right.
And so what we use this for is an adjuvant.
Right.
So we know we need adjuvants for not all vaccines, and that'll be important, but only for some of the vaccines in which they induce a poor immune response on their own. So we add this in to induce the immune response. And so this adjuvant increases the immunogenicity or the ability to induce an immune response. And there are certain vaccines that have this and certain vaccines that don't. An important point is that the attenuated live vaccines do not have this, and they're, in fact, the ones that people have the most fears about vaccines and correlating with various different diseases that have, with papers that have been retracted and discredited that we can go on and on. We have in previous episodes. But yeah, so if you look at a few studies where they look at the pharmacokinetics or the uptake, it looks like yes, you are correct. When you inject a vaccine, you get like a little blip in the amount of aluminum salts that you get, or alum. But the amount that you get from the environment is nearly the same. So if you look at accumulation over time, there really isn't a huge effect that amount of the adjuvant has on the overall bioaccumulation in an infant. I don't know if you have any additions to that or thoughts.
So I have a wonderful book called Vaccines in Your Child by Paul Offit.
Yes.
And it's great because it's written in very straightforward language, and I have a PDF, and he has told me I can give it to anyone. So if you would like it, let me know. But here's what he says. It's actually by two people. Here's what they say about aluminum. So aluminum has been used in vaccines for 80 years. So we have a lot of time to have evaluated it. There's also a very good page on the CDC website.
Yes.
And he says the amount of aluminum in vaccines is far less than a baby will face every day. Aluminum is the third most abundant element on Earth, and it's in food and water. Food is the most common source of aluminum. It's also in teas, herbs, and spices. It's added to leavening agents. It's pancake mixes, flour, baking powder, cheese, cornbread. It's everywhere. And we eat between 5 and 10 milligrams of aluminum a day. So do babies. And they get it in their breast milk as well. And they get way more than aluminum that's in vaccines. So the idea that there's a more concentrated administration of aluminum in vaccines is not really true. It's much less than you pull in your food. Now, a lot of aluminum is bad, but that's way more than you would get in a vaccine. And premature infants are really harmed by aluminum that is present in intravenous fluids. People on dialysis who get a lot of aluminum in the antacids that they're given have problems. So certain risk groups have problems. So antacids, which are given to some patients, have 350 milligrams of aluminum per teaspoon.
Wow. And
that's way more than vaccines. So people have done a lot.
You're talking micrograms in vaccines.
Yes, micrograms. All right. So there have been many, many studies, and this book by Offit has lots of references of some of these studies at the end. So as I said, if you would like that, we can provide that to you. So there's really zero, unless you're in these risk groups, kidney failure, premature infants. There's no harm from aluminum salts.
Right. And there are none in the live attenuated vaccines, like the measles, mumps, and rubella vaccine, which is the one that people have a lot of fear about.
Yes, that's right. Only the inactivated vaccines have them, yeah.
That's right. So there's no reason not to get the measles vaccine.
Right.
So this anonymous writer had a second part to their comment here, and that was the CDC says that you should not administer the second or third dose of certain vaccines to children if they've had an abnormal reaction. Now, she raises a good point here and says, yet the CDC recommends up to 6 shots at the same time, 3 of which can come in a combination vaccine, like the measles, mumps, and rubella vaccine, right? So how is one to discern which vaccine caused the reaction in this case? So in other words, to rephrase this is if you take your child in and they get their 6-month vaccines with like 8 different, vaccine components in 5 or 6 different shots, how do you know if they have a reaction, which one it is, so that you don't give that one again? Right. And it's something that I hadn't necessarily thought about, because when you think about an immune allergic reaction, you don't want to give the same one, but yet you don't want to leave this baby unprotected. So how do you know which ones to do? So I went and looked it up about what are the recommendations for physicians when they have a patient who has a reaction. And so this is from the Advisory Committee on Immunization Practices. And so they say for an individual patient who has experienced an immediate reaction to immunization, it's important to identify the type of reaction, obtain a history, and try to identify the particular agent responsible. So that's what we're trying to figure out, right? So they say that there's an algorithm that's been published, but what they recommend is that they refer them to an allergist, and that the allergist will try and identify which component of which vaccine caused the problem by doing an allergy testing. And then you don't give that one again. You give the other vaccines. So if they had a reaction to Haemophilus influenza B, you don't give that one. Or if you find out they have a reaction to egg, then they don't have any with egg in them. So you can narrow down which component of the vaccine and of which vaccine a baby had a reaction to, and then you can eliminate that from future vaccinations. But you shouldn't leave the baby unprotected. So it's the idea here is if you have actually a severe reaction, understandably, you would not want to go back and revaccinate, but it makes a lot more sense to go to an allergist and figure it out, because You might contact something later. Like, for example, what if it is egg?
Right.
And then you go to feed the baby an egg later after they start eating solid foods, you're going to have a big problem. So it's best if they have a reaction. And when we're talking an allergic reaction, we're talking a severe reaction. And the physician would be right there. They would be able to administer supportive care. These are extraordinarily rare events. Like, you're talking a severe reaction to a component that a child has developed an allergy to.
Perfect. Great.
Perfect.
Very
yeah,
good.
that's great.
Very good. Steph, can you take the next one?
Sure. Niraj writes, Dear Immunosomes, I know it's been a while since I last wrote, but rest assured that, like a lot of folks out there, I am an ardent downloader and follower of the Immune Team's podcast. It is always fun listening to such intriguing stories, and in line with this, the paper discussed in Immune 20 was particularly interesting. As someone who spent years working extensively on inflammasome activation during my postdoc years, this story on the effect of activation leading to blood clots was totally fascinating. And yet again, goes to show that there are still several survival mechanisms that are highly controlled and regulated by the innate immune arm. By the way, I was also a close witness to the discovery or anti-discovery of Cas1 knockout mice actually being caspase 1 and caspase 11 double knockouts, which was pretty intriguing, to be honest. We brought that up earlier. Since those days, a lot of cool science has been done to show the role of caspase 11 in cleavage of gas-dermin D and generation of membrane pores that release inflammatory cytokines like IL-1 beta and IL-18. One interesting result that comes back to mind is that one of the inflammasomes, NALP3, is generally not expressed in primary macrophages, and its expression gets induced by PAMPs like LPS, although I think that was only shown in vitro activation only, which makes me wonder, how is the EPRJ protein alone activating the inflammasome? Now, so going back to Immune 20, I think that they brought that up, that there were other activating proteins. I have to go back and look at that paper. Cindy, I think that was your paper.
It was my paper. Yeah. So I know that there are, I think there are some other things that will both induce the expression of NLRP3 and activate the caspases to induce cleavage. So I think that the I can't say it either. The EPRJ protein probably is doing both. How, we're not exactly sure yet.
Right. And so pontificates, maybe it induces expression of the NLRP3 protein. Right. Probably. And one question I always have is that even though there are several inflammasomes identified, NALP3 seems to be the most robust and universal in mounting a protective immune response against most proteinaceous PAMPs. I know we shouldn't ask why questions in biology, but maybe something we're speculating on. So please keep up the fabulous podcasting, and I look forward to never getting immune to listening to them. That's cute. Yeah, I mean, I think that because, well, one, like they mentioned, there are several inflammasomes, but I don't think we've really looked into the interactions. I'm sure there's ones we haven't discovered yet. What we can say is that NALP3 is very robust, but the why, it's a great question.
Yeah. So I think that there's a lot of different NALPs,
right.
And not a lot of different NLRs in general. And I think one of the reasons why this one seems so central is because there's something related to perturbances that will activate it. So lots of different things like disturbing the lysosomes or ATPs or things like that will end up activating NALP3. So the repertoire of things that we have found that activated are quite broad. And so a central one stimulus is not clear whether all of those things that we have found that do that actually converge on one signal. I don't know. In fact, I think, if I remember correctly, one of the things that activates it is actually relevant to the previous question, which was the alum salts. So the alum adjuvant will cause the lysosomal disruption and activation of NALP as part of the way it works. If I remember the literature correctly. So I think that there's a lot of different stimuli of this that have been found, but whether they all converge on some central signal or not, I'm not sure if it's really clear yet.
All right. Do you want to wrap up with some picks?
Sure.
Yes, sure. So I just want to draw attention to something that's been in the news for a little while, but there's been some updates. So the University of Alaska is facing massive cuts by their new governor. His last name's Dunleavy. And really the general consensus of this is trying to make on promises to potentially reduce taxes by attacking the higher education system, because of their, the fiscal crisis in the state. And, you know, when you think back to why Alaska has this issue. I mean, I won't even go into the depths of it, but it has a lot to do with oil companies. And now that oil, is decreasing in that state. So the state's oil wealth, there's not as much as there was. And so this governor, they're going to restructure, they're going to cut costs to be able to accommodate for the dip or the decrease in revenue from oil companies, which, by the way, got huge tax breaks to be able to drill oil in that state without any, really much putting back into education. And so what they did, I think there was a lot of pressure because it would have been $70 million or $135 million was the original budget cut. But today or yesterday was announced that it's going to be halved. So now the cuts are only $70 million over 3 years. And I think what this represents and is certain administrations attacking higher education by cutting their budget. And I don't think that we can have strong states and strong science without publicly funded institutions. And really these are the institutions that they are the ones that are able to give education to the broadest background of people who come from all different walks of life and who maybe can't get into, it's an easily accessible way for people to get educated. And I just think that paying attention to this, because it is Alaska, I don't think it got as much coverage in the beginning, but now has got a lot more focus. So I just want to highlight that if people should follow. And there's a scientist on Twitter. Her name is Dr. Kat Milligan-Muir, and she's at the University of Alaska, and you could follow her on Twitter for updates. But we need to support even University of Alaska. It seems far away, but this stuff can happen to all of our state-funded institutions.
That's not what you should cut to save money. Education is the last thing that you should be cutting.
Right.
Steph, Cindy, what do you have?
So I have a Nature article, but it's also been in the news quite a bit in the last few days. There are 2 new Ebola treatments that came out that are showing remarkable success. And so you may know that there have been a number of outbreaks, including one that I think is currently going on right now with like 1,200 people have died or something like that, maybe 1,900 over the last year or so. And so Ebola is clearly a very deadly disease. As high a mortality rate is 90%. So they're desperate for anything that will help. And so a trial intermediate announcement was just made on August 12th. And what they showed was that there's basically a 90% protection, which is amazing if they get early enough treatment. And so the preliminary data on like 500 people, so this isn't a particularly small study, showed that there are two different treatments. So one is from Regeneron Pharmaceuticals. It's called Regen EB3. And then the other one is the monoclonal antibody MAB-114. And that was the one that was developed by the NIH, the NIAID. So they've tried both of these, and one showed that 29% of the people given the Regen EB3 died, compared with 34% with a monoclonal antibody. And in contrast, the best that we've ever gotten was 50% of the patients dying with an antiviral drug, or 50% with the previous monoclonal antibody. Well, actually, it's not monoclonal antibody, right? It's a antibody purified from someone who survived the Ebola outbreak in 2014, I think. So this is compared to normally 90% mortality. So you're talking 80% of the people are surviving, up to 90% of the people, if they catch it early enough, are surviving with these therapies. And so it is an incredible move forward. And I think it's going to change the landscape of Ebola outbreaks if we can get these drugs to these people and contain these outbreaks earlier, because I think it's becoming more and more of a concern because there's more movement between populations, more than there used to be, I would say. When you have an outbreak, it used to be contained because it was village specific, and now people are trafficking to larger cities. And so there's a concern that you could have a major, major outbreak.
Yeah, one is a triple monoclonal and the other is one. Yes. So this is great. The only thing is you have to give these intravenously. So you'll have to bring these people to the treatment centers. And seeing as though we're having trouble vaccinating people because of the political situation in the DRC,
yes,
this is going to be
true.
Tough. I mean, it's great, but you have to get it to people. You can have great vaccines. You can have great therapies like this, but if you can't give them to people, they're not really useful. Right?
If they can get it to the medical staff that are helping, that will help too.
Yes, the staff would be great. That would help them so they don't die and they can take care of people for sure. Yep. So my pick, I found when I was looking for a figure last time for Last Immune, a figure of an immune cell, I found this website, Blausen.com, and they have wonderful videos of different aspects of immunology, and you can look about vaccines, corticosteroids, poison ivy, myeloid leukemia, all little videos with nice explanations. 104 different videos on immunology.
Wow.
This is so cool.
Could be useful for teaching or educating yourself, and they'll have lots of great images as well. So the cell in our logo is from Blausen Medical.
Yeah. They have herpes virus, allergic rhinitis.
Yeah, cool stuff.
Oh, neat.
Sorry.
Love it.
Immune 23, immune@microbe.tv if you have questions or comments. If you want to support us, microbe.tv/contribute. You could use PayPal or Patreon to give us as little as $1 a month. We would really appreciate your support. Cindy Leifer's at Cornell University on Twitter. Cindy Leifer. Thanks, Cindy.
Thank you.
Great job.
Thanks.
Steph Langel is at Duke University. Stephanie Langel on Twitter. Thanks, Steph.
Yeah, thanks. This was so fun.
I'm Vincent Racaniello at virology.ws. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.